In early prophase, the centrioles divide, and with asters move apart, the nuclear membrane begins to disintegrate. In late prophase, the centrioles and asters are at opposite poles. The nucleolus and nuclear membrane have almost disappeared. The doubled chromosomes and their centromeres attached to the spindle fibers line up at the mid-cell in metaphase. In early anaphase, the centromeres split. Half of the chromosomes move to one pole and the other half to the other pole. In late anaphase, the chromosomes have almost reached their respective poles. The cell membrane begins to pinch at the center. The cell membrane completes the constriction in telophase. Nuclear membranes around the separated chromosomes. Mitosis is completed. Homologous chromosomes pair to form bivalents. The centrioles divide and move to opposite poles of the cell. The bivalents duplicate to form tetrads of 4 chromatid groups. The nuclear membrane disintegrates, crossing over occurs. In metaphase 1, the tetrads attach to spindle fibers at their centromeres. They line up at mid-cell. In early anaphase 1, the tetrads separate and their paired chromatids move along the spindle to their respective centrioles. In late anaphase 1, the chromatids have almost reached the spindle poles. The cell membrane begins to constrict. In telophase 1, nuclear membranes enclose the separated chromatids. The cell membrane completes its constriction. The first meiotic division ends. There are now 2 cells, each with the same number of chromatids as the parent cell. Prophase 2. In the second meiotic division, homologous chromatids do not duplicate but merely separate. In metaphase 2, the chromatids line up at the mid-cell. The centrioles and asters are at the poles. A spindle has formed. In anaphase 2, the now separated chromatids approach their respective poles. The cell membrane begins to constrict. Telophase 2 has been completed. There are now 4 cells, each with half the number of chromosomes of the parent cell.